I'm Adrian Elbert. I'm working at the provincial medical genetics program in BC Women's and Children's Hospital. What kind of started us on this study, this journey with our colleagues at Virtual Geneticist is really this clinical problem that we had where we would have a patient where, we were certain they have some sort of genetic syndrome based on their phenotype, but the report would come back negative. And we'd always be stumped, how can this be? Is it possible, that there is some number of patients where this is a false negative in fact due to some sort of analytical pipeline issue or human thinking errors in human thinking that lead to the diagnostic variant or variants not being reported. And so you just have this report and it's negative, and that was sort of the end of the line. And so with Dr. Neil Burkle, my colleague, who's also out at BC Children's and Women's Hospital, we went to our colleagues at VG to sort of help support us with this question and see if there are some cases that are of negatives that have the variant in that VCF file, and we can just reanalyze and find a diagnosis in those individuals. What was great was, we often wondered, did we not provide sufficient phenotype? So VG lets us play with the phenotype, which I'll show you. All of the labs have their own non-transparent way of ranking the variants. And so this allowed an alternative method of ranking variants potentially, and those pros and cons of different pipelines are not known. So to kind of give a background, most of you will be aware that with exome or genome sequencing, thousands of variants are detected and then must go through filtering or prioritization tools based on allele frequencies and predicted or previously established deleteriousness, and then patient phenotype gene disease matching as well. And when they're prioritized, we're left with a few, hopefully just 10 or less, that look like something that a human being could actually handle. When the diagnosis is not a slam dunk, for example, there may be a VUS, a hot VUS in question with the phenotype, reporting of variants by the lab can be a bit subjective in the sense that it does require this balance between providing diagnostically helpful variants and avoiding the burden of further workup and counseling of non-diagnostic variants. And so we always wondered whether for some patients the diagnostic variant was below the reporting threshold, and if we could just see the data, could that actually help us? So we hypothesized that there could be many factors that lead to errors in human thinking or low variant ranking by a given laboratory, leading to non-reporting of the diagnostic variant. And so getting that data and immediately reanalyzing it. So not looking for, the evolution of knowledge contributing to new disease genes, but actually looking for diagnostic variants in the most recently performed tests by reanalyzing it immediately after a negative report was what we set out to do. We obtained the raw data for 71 unsolved patient data sets from our provincial program. That's the one in pink there, the clinical cohort. And then we collaborated also with a research endeavor, the CAUSES study, which was a local study that set out to solve more than 500 patients with rare disease, and that's the orange box there. What was helpful there is that they had a diagnosed cohort that we use sort of for our own learning of 269 patients, most of which were done through exome. So the purpose was to answer the following question: whether there is actually a false negative rate to exome sequencing and reporting that could be mitigated by us internally reanalyzing it as clinicians and kind of removing some of that lack of transparency as to what ends up on the report. And then further, whether these AI-based variant prioritization tools, for example, like Virtual Geneticist, could identify misdiagnoses unrelated to new gene discovery. We wanted to thirdly understand the factors that may have contributed to the diagnostic variant not having been reported in the first place. We used Virtual Geneticist along with two other tools for variant prioritization. So those are on the bottom there, the two other tools being Exomizer and Lyrical, which are well-established variant prioritization tools. Looking at the kind of training cohort there, in blue, I'll start off with those results. Just to say that VG was extremely efficient in running all of these samples. So, each case runs for about 10, 15 minutes. And then 20 cases, we could run them in parallel. And so the totality of all of the cases of 800 took approximately 5 hours. These are for the positive controls, so the ones where there had been a diagnosis, and you can see here in blue, virtual geneticist successfully ranked the diagnostic variant or variants in the top number one rank 79% of the time, and the others, it was in the top 10, 99% of the time. This exceeds the other two tools, Exomizer and Lyrical's performance on the singleton analysis. The two misdiagnoses here were within the top 20, and they were actually suspected of only being partial explanations for the phenotype, which may have led to why they were not ranked as highly. Surprising to me is actually the singleton versus the trio analysis did not make a very big difference in the ranking of the diagnostic variant. Although for us clinically, segregation, of course, is helpful in further establishing it as a diagnosis. But the AI seemed to know, which variants were the diagnostic or suspicious variants without knowing whether it's de novo or inherited or in trans, etc. In green, I compare this to Exomizer, where we did see a modest improvement on trio analysis compared to singleton. I also learned through this process as a clinician, the trio analysis wasn't without some pitfalls, including, we ended up filtering out inherited variants from parents that were shown to be mosaic. So you do have to be careful and understand how to use the data. And then there was also a case of one individual's variant being filtered out because of uniparental disomy. So it was homozygous in the proband, but that didn't follow segregation rules. So clinicians do need to be aware of these potential pitfalls when they're using VG on their own. We next looked at undiagnosed cases. So there were several patients in the causes research cohort who had been reported with a hot VUS. And what was helpful was actually looking at VG and how VG ranked these VUS in this separate analysis. So we could see that 51% of those hot VUS were actually were ranked top variant as well in VG, making it concordant with our VG analysis and much more suspicious for me as a clinician that actually that probably is truly the diagnosis for that patient. Whereas in other cases, we could see it's discordant or uncertain, in which case, we could look at the other variants that were also found or listed and ranked. So that was really helpful for me to kind of provide a context for the VUS that was on the report for those patients. It also helps, when it's negative. So this is a case where it's not a VUS that was concordant, it was a discordant one. And you can actually see that by running it through a separate analysis, VG ranked this variant that was on the clinical report from the clinical cohort as number 61 and Exomizer as 25. So making it much less suspicious to me, because now there's this context of, okay, this is somewhat analysis dependent that this variant came up at all, and not so suspicious as if it was highly ranked by multiple tools. In overall, we looked at new diagnoses, and to summarize the new diagnoses in these various arms of our research groups, we had the 24 previously reported VUS with concordance in VG, and one additional new dual genetic diagnosis recognized. Of the discordant, we identified 6 new diagnoses, which are partial explanations. So we suspect that those are twosies. Even in the training cohort, there were 4 additional diagnoses. So those are dual diagnoses as well. And of the negative research group, there were 9 new diagnoses that some require further workup to establish. In summary, we're blue and green represent the research cohort here, and the yellow is the clinical cohort. There were new diagnoses in 2% of already diagnosed probands, so additional dual diagnoses, and then 8% of the undiagnosed. And then in the clinical overall, approximately 10% new diagnoses, which is important because these are, again, in the clinical cohort, not related to the evolution of knowledge, but rather a reanalysis that had the same phenotypic data and did not have additional time passed to recognize that diagnosis. So that's to say that, the false negative rate of clinical exomes is around 10%. And really, this ability for us to look at the data immediately after getting a non-diagnostic report was helpful from the clinical perspective. So, what is our understanding of why diagnoses were missed? One is, there's a failure in interpretation or integration of data relating to mechanism of, when there's multiple gene disease relationships. So that's not really well annotated, whether it's loss of function or gain of function, and that often requires a manual review. But the AI tools potentially are better at those than we previously thought. Similarly, the interpretation of the variance mechanism itself, and then a failure of parsimony. So looking at a single unifying diagnosis in the family when in fact we didn't, or when there's actual multiple diagnoses in an individual explaining the phenotype, as well as incomplete penetrance, and then finally the threshold for reporting. So, whether there was just a single allele in a recessive disease that matched well, that was somewhat subjective, I think, by the clinical reporting labs, whereas we are able to look at those and clinically determine whether we think that might be what we're dealing with in a given case. As noted, there were an increased number of dual genetic diagnoses, probably more than typical, so about 20% in our clinical cohort after this work. And we suspect this is somewhat due to us being the provincial center with more complex patients, and also the fact that they're negative might in fact be because there's multiple diagnoses in a single individual creating a blended phenotype. So we might be enriching for those cases in our undiagnosed cohorts. And then, we had really good success with VG still ranking in these cases both conditions highly. Of course, we don't know what the denominator for that is, but in certainty that the AI is probably doing a better job than humans at identifying multiple conditions in the same individual. What was really great was we are able to play with the phenotypic terms and sort of, parse that out a little bit with VG. So you can submit phenotype that's basically unexplained by the current gene to see if it changes the ranking and identifies another diagnosis, or other kinds of terms might influence the ranking that are sort of synonymous. So I gave you an example here. We had one case of SAMD9 related Mirage syndrome, and the variant is a recurrent variant, de novo in the proband. If you, submitted this HPO set for the phenotype, it ranked pretty low. And this is mostly because this patient actually did not have any cytopenias related to this condition. So it was quite atypical presentation. If you gave fewer number of HPO terms in a set, we actually got a higher ranking. So this sort of change in ranking as related to phenotype, we have to explore these, and we don't know what's going on in the clinical lab when they're extrapolating our phenotype based on our clinical notes. And so I thought this was very valuable as a clinician to be able to sort this out myself and see, okay, like what else, we haven't found a diagnosis, we change the terms, do we get a different list of candidates, etc. And really all to say, as a reflection that there are different types of negatives, and that influences our genetic counseling for the patients that do come with a negative report at the end of the day, or at the end of, their exome sequencing. So it does depend somewhat on the prior suspicion for a genetic condition, where I know, okay, if I was really suspicious, for example, multiple congenital anomalies, consanguineous family, that might lead me to think this is more likely to be a false negative. And also, of course, there's going to be changing phenotypes. So if it's a young patient, we might not have all the HPO terms to get that gene ranked highly, and reanalysis from that perspective might be helpful. And then it was also helpful to look at the data for those negatives, and if I saw that truly I didn't see any candidates in VG on our reanalysis, then perhaps that's the patient I send for genome sequencing to see if we can identify something, or other technologies. Now we can counsel our patients that the clinical lab does have a false negative rate that's probably due to analytical as well as human thinking errors, which is something we didn't really understand as much as now. So the takeaway really is that immediate reanalysis of non-diagnostic data through alternative platforms is beneficial in non-diagnostic testing. And looking at the data by the clinician allows us to value the negative results, truly negative, or to identify potential other candidates or prioritize the patient for other technologies. So just going back to the questions we had posed at the beginning, can internal reanalysis of unsolved cases improve diagnostic rates? We found that yes, so we were able to identify an additional 10%, a diagnosis an additional 10% of those negatives. Can an AI-based tool help identify misdiagnoses? Yes. We found many more purposes actually to using the tool than just identifying misdiagnoses. So we are able to look at the phenotype in different terms, as well as contextualize reported VUS and contextualize non-diagnostic results. So identify negatives as negatives. And I talked a little bit about what factors lead to the misdiagnoses. I think these are areas where, we often see the manual curation becomes really important, looking at those papers. What is the mechanism of a specific phenotype disease dyad? What is the mechanism of a specific variant where there can be challenges in the pipelines of existing clinical labs that we just don't know about. So that was really helpful to see the data for ourselves. And with that, just acknowledging, Dr. Laura Lee and her team that was helpful with the virtual geneticist collaboration, those that were working through the causes study, and then the clinicians at our center that provided the patients for our study, and the patients and families themselves. And I'm happy to answer any questions from our experience.
I just wanted, that was very interesting. I was just wanting to ask, I think you probably introduced this right at the beginning, and I must have been jotting something else down, but when you did this study, this was in the context of a research study, or you were just doing this with your unsolved clinical cases?
We do have a clinical cohort with clinical patients that were unsolved that really became sort of like a QI type endeavor. And then we did have a previously established research cohort that are also existing patients. But those were really helpful because they had in their cohort diagnosed patients that we could use for our own learning around the tool and our own kind of understanding of how VG performed and how it performed against other tools like Exomizer and Lyrical. But it was really clinical in the sense that variants that were identified by us as clinicians, we reported back either to the MRP of that patient, or if it was our own patient, we could do further workup on it if we felt that it needed to be. Like most of the clinical cohort, for example, are singletons, so we would explore, segregation of a new candidate diagnosis and that kind of thing.
Yeah, I mean it's a high enough yield that it almost sounds like something that could be worked into the clinical algorithm for labs immediately, immediately upon getting an undiagnostic exome, that it's like a next tier.
And I think like part of our question is we don't know what the labs are doing on their end. We don't know if the clinical lab is, switching up the terms, for example, if they're getting a negative um, top 10, if the molecular genetics can only handle a certain number of variants to look at. So we don't know if that they don't find any candidates, do they then go and switch up the phenotype provided to their tool or not? I know for our lab locally, um, they're only able to sort of click and analyze once. They have to pay for each time they sort of click analyze, which means that the phenotype that you provide, you only provide it the one time and there's no playing around with it. So that was really something nice with VG, that you can play around with those terms and see, is anything else coming up? Or okay, if not, I'm still suspicious, then maybe I send that patient for genome sequencing or that. Okay, so Steve's question is, I was wondering how the phenotypes were described in what detail. So for the comparison, because Exomizer and Lyrical only take HPO terms, we we used HPO just so we could have an even comparison between the different tools. But I think, it's important to note that virtual geneticists, you don't need to put in HPO terms. It can just use any terms and do kind of like a fuzzy matching because it is AI based. So it is able to, take in different kinds of clinical terms that were, you might not have an HPO term and then try to match with that. So